So when we talk about AIDS, right? So as I was talking about the different perspectives that we have around us, so the major reason is being how it transmits. One is sexually transmitted disease, and the second I told you any chance of body fluids getting mixed up from a person who suffers from this particular disorder to a healthy person, then the healthy person gets a disease. When I talk about body fluids, it excludes saliva, like blood, semen, okay, or serum, or even sexual contact. So these are the ways in which a person can get AIDS. So when we talk about the disease AIDS, okay, just give me a second, another second. Yeah. When we talk about a disease AIDS, right? So it's quite different. Why it's quite different is because the term acquired immunodeficiency syndrome means it shows multiple symptoms or group of symptoms. So how do I explain this? In a very simple manner. You take malaria and you take dengue and typhoid. So when we talk about typhoid, we mentioned the peculiar symptoms, right? We told you abdominal pain, loss of appetite, okay, and then you might have fever, then you might have dysentery or vomiting sensation. So this for this. When we talked about malaria, we talked about cyclic fevers, how it it goes very high and again it comes down, again it goes very high and it comes down. So these are separate symptoms, and this happens when a particular pathogen infects you and the disease shows a specific symptom. Whereas when it comes to AIDS is a syndrome because imagine a person showing both typhoid symptoms and malaria symptoms and dengue symptoms together. So that shows a situation of a syndrome. Understood? A group of diseases. And that is because of the deficiency in the human immune system. There is a severe deficiency in the human immune system. That's what causes it a syndrome. Understood? So the causative agent for the syndrome is human immunodeficiency virus, or it is also called as a HIV virus, a very smart virus. I call it very smart because the way it manipulates our biology and our biological defense mechanism to make us believe that I am not somebody from outside, I am one of you. And then it takes control of our very defense system, right? So that is what makes me very astonishing about this particular virus, human influenza or immunodeficiency virus. So when we talk about HIV virus, it is a retrovirus. Why we call it as retrovirus? Because it has an RNA genome. RNA genome means if you take the structure of the HIV, or if you take HIV virus, the HIV virus like this, okay, has a capsid within it. A capsid is like a capsule within the virus. Within this capsid, what we have is a single stranded RNA. Okay, so single stranded RNA and some of the proteins such as reverse transcriptase, reverse reverse transcriptase, and many other factors which enables this virus to survive within this host. Okay, so how it infects, I will tell you exactly the process of infection and the life cycle of HIV. Let's give a minute. So if we talk about the general aspects of HIV, AIDS was first reported in USA in 1981. In 1981, it started in USA. And when we talk about 2023, within this very short period of time, we have India in the second highest position in the number of AIDS patients in the world. First comes China. First comes South Africa, sorry. So South Africa comes first and India comes second, and it is a highly concerning matter about why this disease, in spite of being spread only through a physical contact of this body fluids, it can spread, and we have so much of cases in our own country itself. Okay, so that is a major concern. So in 25 years, it has killed over 25 million people across the globe. So how does AIDS spreads? Four ways. One is sexual contact with infected person, which is very, very common. Next one is transfusion of contaminated blood and blood parts. Third one is sharing of needles, especially the syringe needles. And there is a possibility of an infected mother who would just transmit HIV to the child through placenta. So that also happens. So when we talk about each of these ways to spread, okay, so we have a specific reason for it. So when we talk about the sexual partner or having sexual sex with multiple partners, so this happens mostly because when a person is going and having sex with unknown partners and their multiple partners. So if a healthy person, if two healthy person, a male and a female, if they both are healthy and they have a sexual contact, nobody is going to get AIDS. For an individual to get AIDS, either a man or a woman should be contaminated, okay, or already should be infected by this disease. Let's say we have a man and let's say we have a woman, and this man is HIV positive, that means he is infected. If he has a sexual relationship or contact with the normal female, I'll write it as NF, she is normal female, then this female will become HIV positive in the later phase. So that was possible only when they had a contact with already infected person. Understood? So that is through sexual contact with infected person. Second one is transfusion. So how does, for example, if I have a healthy person and if I donate it to another healthy person, does the healthy person gets AIDS? No. If your blood is contaminated, okay, if the blood is contaminated by the donor and the receiver who is healthy receives this blood, then there is a possibility of HIV transmission. Sharing of needles. When it comes to sharing of needles, there are several ways in this particular scenario can happen. One, because of the poor medical facilities. Back in the days, now it is very hygienic, or to a very large extent in our country. But if you see our primary healthcare centers just around 15 to 18 years back, okay, 15 years back, let's take, give or take, the primary health centers would just sterilize these needles and they would use the needles again and again. They would just use it on and on for the same person, same needle, different patients. They thought that just by sterilizing it, that is, heating up in high boiling water under pressure, would kill this virus. No, it does not. So that is why a large number of patients were occurred during this time because of low medication hygiene. So now we use one needle, one use, right? So once you take the needle and inject a person, it locks in and it does not come out in many of the syringes that we use in hospitals. But we have another problem that is concerning us is drug communities. So there are some drug communities that people make up when they're addicts to a certain drugs like heroin. Okay. So when they are taking these drugs, they have a communal drug abuse where they use single needles for multiple person. Okay. And there is high probability of this HIV being infected. And this happens because they usually lack very high levels of hygiene when they're drug addicts. Next one is from a mother to a child through placenta. So the chances are high because the placental barriers are very high for the first child, and as the go for second and third child, it is great possibility that these kids might not get the protection as the first time. Okay. Hold on. Yeah. Okay, let's go for the next one. So if we talk about AIDS and we talked about the parameters in which people will get HIV and how it is possible. Okay. How do we prevent AIDS? So what happened? This is one important slide I would like to talk. If you just see like what is to happen, I remember very specifically back in the days when around 10 years back, okay, long time, I know that. 10 years back, when I was watching a TV on the news, TV9, okay, I know it's wrong to mention the channel name, but still came out. So in this TV9, they give this case. They did a right thing though. They give this case where a family was thrown out of the village, and these a husband, a wife, and a child, they were living outside the village. Okay, and they were not allowed to buy any groceries, collect water. The child was denied education in that particular village, and nobody would give them house for renting or work for, for them to perform, because of one reason, that is, they thought AIDS is very dangerous, and these people being in the village, by touching them or they touching any other things in the public, would cause AIDS. And the family was highly discriminated and they were thrown out of the village. Now they were facing a lot of challenges as a family to live a normal life in the society. And people had to be educated. And TV9 brought this thing and they told that this is not the case. It's not supposed to happen like this. People do not get AIDS just by merely staying with them. So this was happening. But anyways, villagers were not able to get convinced. And this is what the impact was on that family because of low education. So people who did not get educated or chance to learn about AIDS. We know how it spreads, but we don't know how it does not spread. So how does not AIDS spread? For example, if you kiss even an infected person, you will not get AIDS. Let's say we have two individuals. One is positive, other one is negative. Positive means the person who have AIDS. Negative means the person who does not have AIDS. If they kiss, will something happen? No, it does not cause AIDS. Next one, hugging. Even hugging infected individuals will not cause AIDS. Sharing of food. Next one is insect bites. What? Why not? Okay, interesting question. Why not? When a mosquito bites an infected person and it goes and bites a normal person, AIDS do not spread. Let me tell you. When mosquito bites, it sucks blood. When mosquito bites again, okay, let's say that mosquito goes and bites another person, healthy person. At this point, mosquito will not inject the blood. Mosquito acts like our digestive system, where the AIDS virus cannot come from the blood all the way till the saliva of your, mosquito. So when it bites, basically the saliva is what can get into our human skin, not the blood. Understood? That is why mosquito infections will not cause, mosquito bites will not cause AIDS. If you share the toilets, so I use public toilet, do I get AIDS? No, you don't get AIDS. Next one is bathing. Even if the infected person has bathed together, or if you use the same water, okay, for example, you cannot get AIDS. Next one, sneeze and cough. Okay, if an infected person just sneeze on you, or if you touch an infected person, do I get AIDS? No, you don't get AIDS. Next one, sweat. If you go to a gym and you find sweat and you think that, okay, if I touch an infected sweat, do I get AIDS? No. Remember this. It's very important for your communal behavior. Kissing, hugging, sharing food, insect bite, toilet seats, okay, bathing, or sneezing and coughing or sweat. All these factors does not cause AIDS. Okay, sure. Now, this particular AIDS is so dangerous and so smart. Why? Why? Because when the AIDS get into your body, they can really cheat your body for a very, very long time. How do they do that? If today I make a contact with an infected person, and let's say that this is the day. Now, tomorrow I will not get AIDS. It may, but most cases from 5 months to almost 10 years, okay, 5 months to almost 10 years, you will not be able to recognize any symptoms of AIDS. At this point, what happens is the AIDS virus is in the dormancy. What is dormancy? They go and hide in your cells and tissues for a very, very long period of the time without causing you any infectious symptoms. Like no symptoms will be there. For 5 months to 10 years, you cannot identify, even though the virus is there. So this is called as incubation period. Incubation period may the AIDS will be in an inactive form. Many diseases have incubation form. Most of the diseases have incubation period. But AIDS being such a dangerous disease, it can remain incubated for 5 to 10 years, 5 months to 10 years without you getting noticed. That is why it is so hard to keep track of this disease. That's why you have to frequently go and get checked once you reach adultery. Okay, when you reach 24 or something, every 6 months once or 1 year once, you have to get it checked for AIDS. Okay, so after the incubation period when the infectious cycle starts, and then the symptoms starts appearing. That's why it's so deadly. AIDS is so deadly. Okay. How does HIV cause its infection? Let's say the HIV enters a body, and this entry to the body will happen only through body fluids, blood, semen, serum, okay, etc. So when they enter our body, the major cell they target is macrophages. What are macrophages? Macrophages are a type of immune cells which look like amoeba-like cells. Okay, they look like amoeba-like cells. So they attach to these macrophages and they initially increase their number. How do they do it? They get into the macrophage cell, they attach to the macrophage cell, they release their capsid that I talked about and the single-stranded RNA. So this single-stranded RNA will be released into the cell. The single-stranded RNA is converted into DNA. RNA to DNA, RNA to DNA is called as reverse transcription. So that is the enzyme. Reverse transcriptase is an enzyme. So that will convert the RNA into a single-stranded DNA. So this viral DNA will get into the nucleus of the macrophage and it will become a part of host DNA. So when it becomes a part of host DNA, you know that their software is hacked now. So the DNA of this host is hacked. So that gets incorporated into the host DNA. Now, what is the role of DNA? DNA will produce more RNA. So it will start producing RNA and RNA proteins as well. RNA particles are like RNA proteins, and viral RNAs are also produced. Understood? So this HIV virus that is coming out of these infected cells, they enter into new type of cells called as T cells. Especially they are called as T4 helper cells. Okay. So what are these T4 helper cells? We know that there are two types of immunity. One is caused by B lymphocytes. Other one is caused by T lymphocytes. B lymphocytes is called as humoral immunity, correct? And it produces what? Antibodies. Correct? No. Now T lymphocytes will produce cell-mediated immunity. Correct? They produce what? Cell-mediated immunity. Now what happens, students? There is a bridge between humoral immunity and cell-mediated immunity, and that comes through T4 helper cells. What do T4 helper cells do? They are produced by T lymphocytes. They will actually have the memory of this particular virus. So what they do is, anything, they will have memory. So this memory will be given to your B lymphocytes. Based on the memory, they produce these antibodies. Now imagine that they are hacked or they are attacked by this HIV virus. When they are attacked, the T4 helper cells will decrease. When T4 helper cells will decrease, now there is no proper communication between the cell-mediated immunity and humoral immunity, finally crashing the entire immune system. You see how crazy this organism is. So that's what it does. It attaches to helper T cells, thereby completely destroying them, reducing their numbers. So they replicate and they replicate into new progeny viruses. Now they attach to more T4 helper cells or TH cells. Now the T cells will decrease. Once the T cells will decrease, overall immunity will decrease. And once the overall immunity will decrease, that's it. Okay, the person will suffer from multiple symptoms and it will take a very, very hard time curing this disease. Understood? Excuse me. Sorry. Okay. Do you understand? One second. I'm not able to. Yeah, sorry. Did you guys understand how the HIV spreads, or should I just summarize once again? Students. Okay, done. Others? Okay. And then you're also good. Okay. You have understood, right? So we can go further. If you feel that, okay, I think I need one more time repetition, it's okay. You can ask. All right. So no problem. Anyways, assuming that you people are little awkward to ask, so let me just do a quick revision. Okay, very quick. I swear that I will not eat your head. This is very important. Life cycle of HIV. Okay, so 1 second. What happens? How does a HIV spread in our body? Okay, So simple. HIV enters our body through body fluids, serum, semen, blood. Through that, it enters our body. Once it enters, it attaches to a special type of amoeba-looking like cells called as macrophages. They'll increase their number. I told you HIV has RNA as its genetic material. So this RNA is very well hidden in a viral capsid. You can see here. This is the capsid. Okay, this is the RNA that is present. So now what will happen? This RNA is injected into the host cell like this. They're injected into the host cell. Once they're injected into the host cell, what happens? They will go inside the cell. They will convert this viral RNA into viral DNA. So conversion of RNA to DNA is called as reverse transcription. And reverse transcription is done by an enzyme called as reverse transcriptase. So once it produces the viral DNA goes into the nucleus and it gets incorporated into the host, thereby controlling the infected cell. The infected cell will synthesize RNA and protein. Correct? No. DNA means central dogma. It should follow the same thing. So all the RNA are viral RNA and all the proteins are the ones that support your HIV to produce more and more of this progeny. Understood? So each of them will enter into the cells and they take control of the cells and they produce more of their progeny virus. Okay? So these HIV, it enters into another type of cells called as helper T cells or T4 helper cells. So when they go there, they make them as HIV factories and they produce more of the progeny viruses. So as they enter into more of the T helper cells will decrease or T cells will decrease. When T cells will decrease, their immunity will weak. When immunity weaks, any infectious or agents that are present in our surrounding, like tuberculosis, cholera, anything that we are not getting diseases, even a common cold can be a very, very serious case with respect to AIDS patients. Okay, so they will suffer from multiple diseases and multiple symptoms, so thereby showing the syndrome like character, which is called as acquired immunodeficiency syndrome. Now, what are the effects of a person who is suffering from HIV? So basically they have multiple symptoms. So most of them have mixed symptoms such as fever, diarrhea, weight loss, okay, deficiency of T4 cells, okay, this is T helper cells or T4 cells, okay. Next one is mycobacterium virus, basically TB infection is very, very high, and fungi infections are very common, and parasites like, toxoplasma are also found, which is very rare in normal human beings. That are all will be found in HIV patients. So these are some of the ways in which the HIV changes the body. Next one is diagnosis. So when we talk about diagnosis, we have already talked about this molecular kit called as ELISA, which stands for enzyme-linked immunosorbent assay. So this ELISA kit is basically, they have that is antigen-antibody interactions. Okay, antigen, and then we have antibody. So based on these interactions, now you will get a confirmation whether it is HIV positive or HIV negative. Understood? Now, coming to the treatment, the most common treatment for HIV is use of antiviral drugs, okay, and also partially, just give the treatment for the symptoms. So you have these symptoms, okay, we'll give this type of an antibodies or we will try to reduce the symptoms. So it is symptomatic based, healthcare or cure that we have. Do we have medication for HIV is a controversial subject. Some say that medication is there. Some say that medication is not there. But the medication for HIV that is most effective right now, we can't consider it as 100% working medicine, but it can prevent to a certain extent. And it is very, very expensive. Okay, medication for HIV is very, very expensive. That is why in most of the healthcare centers, they cure for symptoms. So that is why prevention is better than cure, and cure is very, very hard with respect to HIV. How do we prevent AIDS? Just now I told you one story. How people without this basic knowledge of how a disease acts, like how it behaves, how it spreads, is it okay to involve people, not involve people? Without this common knowledge, right? So it will be very hard to understand how to prevent it or how to, approach this particular disease. So that is why we need to educate people about AIDS. We should take it openly. Discussing about a disease is not a sin. It is not shy. Oh, AIDS disease. There is nothing that is very, what do you tell, funny about it. It is a disease. It's a serious condition. It can happen to anybody. So it's necessary that people know about it rather than having a particular perception about it. Okay, you should not perceive, oh, this is funny. These people got AIDS because of that particular reason. No, it should not be like that. Can happen through anyways. So that is why how it spreads, how to prevent, how it does not spread, what you should do when you get find the symptoms. Should you rush to the hospital? Should you get it checked? Yes, of course. All these things will help you to bring this awareness about how to understand and approach this disease. Next one, controlling a drug abuse. I told you, communal drug abuse is a very serious problem. So there's dark groups that people go with, these cults. So they have this community of friends or people there, even though some are smart and some are dumb, because of circumstances, they just take it very lightly and they start sharing needles. So that is very, very a high ratio is there where the people can become HIV positive. Next one, medical centers and personal use. Never use one time needles, sorry, or never use repeated needles. Always use disposable needles and get rid of it as soon as possible. And advocating safe sex in the sense, first one, know your partner's health. Avoid multiple partners and unknown partners, especially for sexual contact. So that is far better. Things can get lighter and things can go real bad. That's why. Next one is free distribution of condoms. Government has done its part in educating people about the role of condoms in preventing HIV or some of the serious disease like HIV. So condom use can prevent it, and distribution of free condoms is very nice. Next one, regular checkup of HIV. As I told you, since it has a very long hibernation period of 5 months to 10 years, it's very essential that you get checked up regularly without shy. It's fine. Okay, health is better, prevention is better. You might be 1000% aware, but still you don't know, right? Incubation period, you can happen anything. So we don't know. We can't be 100% aware all the time. That's why it is highly essential and advised to get it checked at least once in a year. Next one, making regulated blood banks and surgical instruments or any of this medical equipments like dialysis machines. So very well checked and very thoroughly inspected before the other patients are subjected to a particular treatment. Understood? So these is how we prevent AIDS. It's very common knowledge, but if you just look at it, it will really help us to understand it. Okay, is it clear, people? Did you understand? Yeah, can we go further? Yes.
Yes, sir.
All right, thank you. Let's go for the next topic. And our next topic is cancer. Okay, so cancer, everybody knows about cancer. You might have heard about cancer, and cancer is bad, and it is very, very bad, and it is a quite a disease to be studied. Why? Because cancer is very, very much heard about in our society, and many of the people suffer from cancer. Some people, even getting the start of a cancer, they fear for their life. Some people who are in a very serious condition, they know that it is certain that they will die, yet you can see them smiling and enjoying their life, enjoying their last moments. That is why the knowledge about cancer is quite important. And cancer is an abnormality that can come to anybody at any point of time for several reasons. Even though you are like completely proper with your health and your diet, just because of some mutation or your genetics, you can still become cancerous patient. So what is it generally? How does cancer? What is cancer? Cancer is an abnormal growth or uncontrolled multiplication of cells. So whenever you talk about your body, that's why I tell you, when you look at the biology of your human body, it is beautiful. Why I tell it is beautiful is because They are so well regulated in the number of cells you must have. If you look at a person's eyes or face or any muscles, you don't look at them deformed. Hands will look like hands, eyes will look like eyes. So that means they accumulate a particular shape, particular number, particular texture, particular layers. So that is how we have this unique and uniform characteristics throughout, from one generation to other generation. So when you look at humans, you like human-like characteristics or human characteristics, correct, right? Whereas imagine, for this to be accomplished, your cell should have such communication and such control over its own growth. Correct, right? So that is why our cells are, each and every cells are fitted with this time, like a time bomb or a timer, like an alarm. So when the cells divide, okay, this alarm starts and they start growing, this alarm keeps on ticking. So when the cells come to an age, they divide, and when the cells have reached their time, or when the cells have reached their limit, the clock stops, and finally it will make the cell to arrest its cell division. It will tell it you can no more divide. So that is a strange behavior that our cells goes. Now, once upon a time, this cell had the capacity to division, and now it has lost its capacity to divide. How did it happen? It had the capacity to divide or not divide is a character, and that character comes through genes. Even for the cell to tell it to divide or tell it to not to divide, we have genes. And those genes are, one is oncogenes, next one is proto-oncogenes. Oncogenes means oncology, onco, cancer. Even though we know that it defines to cancer, it's not a bad gene. This oncogenic gene should get on so that it promotes your cell, it tells your cells to divide. Proto-oncogenic genes, okay, or tumor suppressing genes, what they do is they act like brakes. So these brakes, what they do is they will tell the cells to stop this, cell division at a certain point. These brakes can be activated based on the cell type, cell's function, cell structure, everything. So many characteristics will come under this. Now, if the brake fails, what will happen is the cells will start dividing and dividing and dividing without any control, leading to the formation of a mass of cells. So this is called as a tumor or a cancer. Understood? So it's all genetics. Now, usually, when we talked about the cell's capability to recognize how much I should divide and how much I should not divide, that is called as contact inhibition. When normal cells divide, they know that exactly how much their division should last up to, or when they should stop the division. So when they have reached a certain extent, that means that they are able to sense something. What do they sense? They sense their neighboring cells contacting them or coming in contact with each other, where they communicate with each other and they come to a conclusion that we have reached this much of communication or bridge. That means it is the time to stop our cell division. That is called as contact inhibition. When they come in contact, their touch itself is a communication to stop cell division. So that is shown in the normal cells. So if this capacity is lost in our normal cells, then it becomes cancers. So they don't care how close you are to the next cell. So they start proliferating where they lose this contact inhibition. So that leads to uncontrolled growth. So when somebody asks, does cancer cells have contact inhibition? You should tell no. Because they have lost the property of contact inhibition, that is why they are able to divide. Okay, understood? So when we talk about cancer, cancer can be divided into two types. One is benign cancer, other one is malignant cancer. Localized cancer, non-localized cancer. When we talk about benign tumors, okay, or benign tumors, what happens in benign tumors is, see, in benign tumor, a particular layer of the cells, they get cancerous and they start proliferating to form a tumor or a mass of cells or tumor mass. Once they have formed this tumor mass, they are localized to this particular region. If it is intestinal cancer or colon cancer, they are in colon only. If it is oral cancer, it stays in oral cavity only. So this is called as localized cancer or benign cancer. So they remain to that part where they are affected and they do not spread to other parts. Okay, and they are quite damaged, a little bit of damage to the tissues. Next one is malignant tumors. Malignant tumors are quite dangerous. Why? Because once we have the cancer cells, okay, so these cancer cells of malignant nature are called as neoplastic cells or tumor cells. So these neoplastic cells or tumor cells, they have grown into a certain mass in a particular region. Now, what happens to them is they disassociate or they get detached from the original site where they were causing the cancer. Now they migrate to different parts of the body. It can be brain, it can be lungs, it can be liver. From bones, it can migrate to brain. Okay, so like that. When they migrate to different parts of the body, they attach to a newer region and they start cancer in the new region. So this is called as metastasis, and it is also called as malignancy. The capacity of these malignant cells to detach and cause cancer in a new location is called as metastasis. Understood? And this condition is called as malignancy tumor. And they're very, very vigorous, highly active in the cell division. So because of their active cell division, they usually are taking a lot of nutrients from the normal cells and tissues. They channel their own blood channels for higher oxygen supply and nutrient supply. They take over this blood supply and starve the neighboring healthy cells, thereby losing you weight, you will become weaker, you will have tiredness, and so many different cancer symptoms. So this is called as malignant tumors. Causes of cancer can come because of something called as carcinogens. Carcinogens are cancer causing agents. So these carcinogens can be anything. It can be physical factors, chemical factors, or biological agents. So, for example, for carcinogens, when it comes to physical agents, is ionizing radiations like X-ray, gamma rays, and non-ionizing radiations like UV rays. For example, let's take UV rays is very important for us. Why? Because we apply a lot of sunscreen and we prevent the sun from damaging our tissue. Let's say that UV radiation is high and it falls on your skin cells. So now what happens? We know that in our DNA, there is adenine, thymine, cytosine, guanine, like that nucleotides are there. When an UV radiation hits, especially on the thymine region, it causes something called as thymine-thymine dimerism. That means two thymines will come together and they break this normal linear polynucleotide chain. Like the straight chain of your nucleotides in your DNA is broken or the structure is altered because of this radiation. So that is when it leads to skin cancer. So example for physical agents, it can be radiations like X-rays, UVs, or gamma rays. Next one is chemical agents. Chemical agents are usually one of the most famous chemical agent is the tobacco. That is, when you use this tobacco, you have nicotine and many other carcinogenic substances are there. Next one is vinyl chloride, caffeine. Okay, and then you have nicotine, that is in the tobacco. Mustard gas are some of the carcinogenic chemicals which can cause mutations in your cells and cause you cancer. Biological agents are oncogenic viruses, cellular oncogens called as C-onc, and then you have proto-oncogenes. That's what oncogenes and proto-oncogenes, and then you have some of the cells, okay, which are activated by these oncogenic viruses will cause you tumor or tumor-like conditions. So these are called as carcinogenic substances or cancer-causing agents. Now, how do we treat cancer? Treatment of cancer begins with detection of cancer. That is why the cancer detection and diagnostics is one of the most costliest and most expensive medical treatments on the world today. Detecting cancer will cost a lots of money and it's a lot of technique also. So now cancer detection can be done by biopsy, imaging techniques, use of antibodies, and then molecular basis of techniques or molecular biology techniques. What is biopsy, students? You go to a cancer region and you pick up some of the cells, okay, or tissues from the region of cancer, and then you grow it in your labs, okay, in the lab when we grow it, okay, and then we examine it under the microscope, it shows its way the cancer cells are dividing. How many cells per hour are dividing? How vigorous is the cancer? All these things can be studied by something called as histopathological studies or histopathological assay. So when we perform this, basically it shows the characteristics of the entire behavior of these cells. Thereby we can detect which cell has cancer. Understood? So that is called as biopsy. Biopsy is taking the piece of or some part or some tissues of this cancer region, and then we grow it in the lab and conduct histopathological assay, thereby studying the nature of these cells and declaring whether we have cancer or not. Coming to imaging techniques, they are very, very useful not only to detect cancer, to understand how big is the tumor, to where it has spread, is it operable, how deep is the blood and tissue integration with this. All these things can be studied through X-rays or 3D imaging like CT scanning, like computed tomography to study brain tumors, or MRI, that is magnetic resonance imaging, which is so useful. You can even nerve conditions or nerve connections you can see through MRI. Okay, hold on 1 second. Yeah, I hope you're understanding. Okay. Next one. In MRI, what we will do, we'll do magnetic fields, okay, and non-ionizing radiations. So when we create this magnetic field and non-ionizing radiations, you basically understand the entire physiology of humans. And here, nerve connections, blood connections, or where the tissues have integrated, everything you can see very properly in MRI. Okay, so this is called as imaging techniques, and they're very, very useful to understand the vigorousness or how the cancer is spread. Okay. Next one. Antibody assays or immunoassays are very, very useful nowadays for cancer detection. A certain cancer antigens are detected on the cancer cells. For example, if we have cancer cells, okay, so this is the cancer cells. In the cancer cells, sorry, in the cancer cells, what do we come across? We come across some surface antigens. What are the surface antigens? So these antigens are produced only by cancer cells and not the normal cells. So anything like that is called a surface antigen. So they are detected using an antibody specifically designed for that. So they go and change, and thereby causing a small indicator. We get to know whether the patient is suffering or not. So this is called as antibodies used for cancer detection. Understood? So next one. Next one is radiotherapy. So radiation, like uranium, right? So similarly, radioactive material is used for your radiation therapy. So the normal cells are given interferons to not to get affected by radiation, okay, alpha interferons, whereas the the normal cancerous cells will be targeted because these cells will not have interferons. So the normal cells will be self-protecting about this radiation, already given the information about radiation. Now what happens is this radiation is exposed to the areas of the person which is suffering, the cancer cells. So when they're exposed to these areas, the cells will die. Tumor cells will start dying because of the irradiation. Understood? So this is about that. And most common therapy is also chemotherapy. So cytotoxic chemicals basically are used. Cytotoxin means the chemicals which kill cells. So they do targeted chemotherapy and general chemotherapy. So slowly the chemotherapeutic drugs are injected into our system. So these cytotoxic cells, they act on these cancer cells. So since they're cytotoxic, that means they're toxic to cells, they'll kill these cells. So as the cancer cells are dying, on the same way, what is happening is even the sensitive cells like the eyebrows, the cells of the eyebrows, and the eyelashes, hair, all of them will become very sensitive to this chemical exposure, and they start falling down. Understood? So that's why the cancer patients go bald during this chemotherapy. It's very sad, but we have to support them emotionally. Okay. Next one is during this cancer patients treatment, they also experience lot of weight loss, weakness, vomiting sensation. They vomit a lot because of the radiations and the chemotherapy. So their appetite is completely gone, and they have too much of tiredness and weakness. So this is a very dark side of the treatment. and it takes a lot of strength for the people to beat it. That's why we should have a lot of respect for the people who are fighting cancer. Okay, so that is about that. Understood. Next one is immunotherapy. This is the latest one in immunotherapy. Usually, patients are given a biological response modulators or modifiers, for such as alpha interferons. So here, what happens is they go and attach to a specific tumor cells, thereby directing our own immune cells to go and eat these cells. Okay, it will make sure that our immune system recognizes these cancer cells as enemy cells, okay, or antigens. So that is when our immune system starts digesting it. So this is the most advanced one, promising one. Understood. So cancer can be treated by combination of drugs like surgery, radiotherapy, chemotherapy, immunotherapy, etc. There is no one specific treatment for cancer. Understood. So it's a multiple treatment. Anyways, we have come to the end of this session. Okay, I'll take the next class tomorrow and we'll talk about the further topics. All right. Did you guys understand? Yes, no? Thank you, Nandan. Yeah. All right. So if you have any doubts, please make sure that you leave your doubts, okay, in the coming class. Others, you can leave, okay, if you have no doubts to ask.